A presentation.
Thank you very much. I should say that, as with several others, my throat is a little bit infected, so my voice may fail at times. There are several things I don't have time to talk to, but there are several things in the back of my mind whenever we framed our research program. And I just mention these because I have so little time to say what I want to say. I don't want you to know kind of what's going on in the back of my mind. The other thing is that in everything that we do, we find it, it's a great book, by the way. I really recommend reading it. In everything that we do, we sort of have this attitude that we could be wrong. Indeed, that all of us could be wrong. And one way around that is to, if we ever have doubts or differences of opinion with other scientists, we run little round robins with them until we absolutely agree on the outcome. So, I mean, I guess that's what that book says, is that there's nobody really needs to be wrong. What matters is that we pursue the truth until we're sure. You're looking at a section of a cell and 40 nanometer particles. This is just live cell imaging. And I'm showing this as a talking point to remind you, I think, what you already know. These are all nanoparticles that are unsuccessfully challenging this membrane. And this is one particle that is in the process of endocytosing. And I mention this just to remind you that all the events that we're speaking about here are energy dependent, they're managed, they're to do with recognition of the nanoparticle. They've got nothing to do with diffusion and partition and sort of equilibrium chemical principles such as small molecules usually have. And that story, I think, is absolutely known, but absolutely important to keep coming back to in all that we do. I'm also not going to spend any more than a few moments in this because you know this very well, I think, except to remind you that the endothelial surface can be several thousand square meters, and there are all sorts of other challenges and so forth. So the point is that what we're asking really is quite a lot when we're asking about active targeting. Because we're not just asking that we reach the site, but crucially, we're asking that we don't reach other sites. And I wanted to focus your mind a little bit on this. Again, I won't spend much time on this because I think you know all of this very well. I'm just commenting on this just to think a little bit about what we're asking, the complexity, the velocities, the relative velocities, the relative volumes, all of these issues worth sitting thinking about to really understand what we're asking from ourselves when we're doing active targeting. Okay, so I suppose a little bit strong statement, but maybe it's worth just sort of highlighting. There's no such thing as an untargeted particle. They're just good targets and bad targets. And this 5,000 square meters of area that you can sort of imagine laid out in front of you. The idea is that I've got to avoid all of it with my particle. And since recognition is at this size scale absolutely fundamental, it's not that easy to avoid all of these things, right? So, I'm afraid my throat is drying up a bit. Just a minute. So the basic hypothesis I want to talk about is really this, that what nanoparticles actually do is not just a consequence of what we make, but how they look in the environment in which they're applied. We have a review of this last December in Nature Nano if you would like to know more details. But to remind you of the sort of basic concept, let me just take an example. So let's take the act of endocytosis. There's nothing special about that particular process. What I want to say is we presume valid for all processes, but there will be a characteristic processing time for endocytosis, for example. You saw that occur in the movie. That's several minutes during which things are recognized, various things are recruited, and all of these details we don't need to go into. And the question is, during that time scale, what is happening to the particle on and in the vicinity of the particle? And the basic hypothesis is that besides whatever you've grafted onto the particle, for example, there are other things associated to the particle that are taken from the environment. For example, if this is human plasma, there'll be other proteins and lipids and so forth. And some of those will exchange slowly and some rapidly. And the basic premise is that those things that exchange more slowly than that characteristic time that is required for processing can be part of the recognition event. So it's not just a question of something being stuck or not stuck, and it's not just a question of one thing or another, but there's a group of molecules that may be associated for a significant time. Now, I'm oversimplifying this hypothesis because besides that, whatever is associated for a significant time to these particles also has to be oriented in the correct way and all of these sorts of things. Now, I start with something that is, I consider well-nigh trivial, but just to say it, nevertheless. I mean, particles that are unmodified, for example, that are not pegylated, I hope that you will one day have exposed them to some biological milieu. I hope that you will have used a disc centrifuge, and I hope that you will have seen that when you spin down the particles and wash them, that there is a shift that doesn't go away, and the shift in their nominal size is due to what has been associated to them. And it is now a very simple and I think not that exciting story to simply determine what is associated to such particles. Essentially, it's a very simple story because the exchange times are so long for all of these typical particles that you know, silica, titanium, all of that. They're so long that really this idea of long time scales and exchange is not relevant. They're just, everything is stuck for so long. And one can presumably, you could actually do quantitative proteomics and find out what's on the surface and so forth. One small thing I do want to stress, though, in this slide, which is not entirely trivial, which you may not remember. If you just look, let's take a very simple particle, silica. As you increase the concentration of, for example, human plasma, what actually is associated to these particles is completely different. So, for example, at 10%, which might be typical of what we do in vitro, the proteins that are associated to the particle bear no relation at all to what they look like in more concentrated human plasma. This begins the sort of whole question that we need to look into a little bit more closely about how we should be working in vitro. How do we make something more realistic? Now, I just wanted to again show you this, again a simple slide. It makes a simple point. These are protein arrays. There's 10,000 proteins spotted in pairs. There's nothing exciting about these anymore. You can buy them in a catalog. Although, if you want to make a liver, for example, you've still got to make it yourself, by the way. But you can spot all the proteins in a human genome, and you can spot a liver, you can spot anything you want. What I'm showing you here is a typical polystyrene particle. There's nothing special about these stories that I'm telling you. And unsurprisingly, this actually provides a very interesting map, by the way, of particles. I don't know if you've ever thought about this. But basically, unmodified particles, for example, in PBS, stick to everything. Now, I just want to mention to you that if you take those same particles and you begin to add, for example, human plasma, then at about 10% really is most uninteresting condition that you could possibly study. It's not until you get very much closer to real plasma that you actually begin to get well-defined targets, which are presumably the biological targets of those particles in that milieu. Again, just to say that the biological targets of the particles is context dependent. It depends what they're in. And I'll hardly even need to say that there have been little errors of understanding by, I don't know if you probably do know this, but if you work in the wrong milieu, either for the wrong organism, or for the wrong animal, wrong serum, mathematics, or if you work at the wrong concentrations, you get physical interactions with cells that are entirely irrelevant. Whereas typically, if you go back to the realistic milieu, all of the endogenous machinery switches on again, and you get back what is sensible biologically. Okay, so we can figure out what's on the surface of such simple particles. We can even predict their targets. And it is barely, I won't spend much time on the slide, except to say that the next little piece of the puzzle is to be sure that what is associated to the particle does, in fact, guide, for example, their endocytosis and uptake, and it does. In these particular particles, for example, it is the LDL receptor that is primarily taking up silica particles in human plasma. It's got nothing to do with the silica, it's only what is adsorbed to the surface and expressed. I won't spend much time on that because I think that is pretty much obvious. You stick any common particle into a biological milieu, it sticks on something, and typically, if you do appropriate silencing studies and so forth, you can soon find out that the way in which it interacts with biological targets is in fact to do with what's adsorbed onto it, not the object itself. And that happens in realistic environments. Now, this is a little bit more subtle. It will have occurred to you, I think, that when you go to low-energy surfaces, for example, pegylated surfaces and so forth, there's much, much less protein adsorbed. And it becomes very difficult to look at physical experiments to see how much is adsorbed. This is in Nature Nano in February, by the way, if you want to look at more details. The point I want to make is that we need to be careful in assuming that in pegylated systems, the story that I've just told you has gone away because we have reduced the amount of bound protein. What we've done here is this is a very simple model study, so it's transferrin grafted with PEG, and we're watching the uptake of those particles, for example, and how they change as we knock down the transferrin receptor. And there's a nice, solid knockdown. I won't go into some of the details when you're in PBS. All of that knockdown vanishes as we go back into human plasma. And furthermore, I would like to stress that this human plasma is completely depleted of endogenous transferrin. So what you're seeing here is not a consequence of competition from the other protein, but is in fact a consequence of being in this complex environment. I'd like to stress here that this does not mean that particles stop going in. They continue to go in, and this is a ubiquitous story, by the way. There's nothing special about the particles, the grafting, the ligands. It's ubiquitous. But what I'd like to stress is that particles still enter, in many cases, for many particles, but they don't enter by the route advertised. And really, actually, I don't have time to go into this, but if you watch over the next year, you'll begin to find what I think is a more interesting part of the story, which is what they do go in by in complex environments. And that turns out to be an absolutely fascinating story, because these are essentially off-target interactions. So as soon as we put these beautiful particles that we believed in and trusted into a complex environment, we certainly lose a lot of targeting. In fact, in some cases, we lose all the targeting. In some cases, we can keep a significant amount. But what we do is generate a whole new set of targets that we're now able to map out in terms of receptors. The reason I'm stressing that is that essentially that's sort of off-target issue, those receptors are somewhere in those 5,000 square meters, somewhere else in the liver. And we have to think a little bit about this. Now, what we've been doing is kind of trying to understand how the control of this interface, the grafting of, for example, PEG and these things, can be used to manipulate all of this. But to stress that what we have to do is to look at all of these effects in a realistic environment and not in either PBS or in a simple laboratory style experiment. So I think loss of specificity in one cell may well spell off-target uptake in many others. And so now what we're doing, as you can probably imagine, is sort of robotic, right? So trying to understand off-target. Just watch that space and you'll see what I mean. The last point I want to make is that the story that I've just told you does not cease at the receptor. Although we understand much less about it, what we've begun to understand, we think, what we've done here is GFP labeled all of the RABs. So we're sort of doing a complete RAB silencing screen and also GFP labeling all of the RABs. So what we're trying to do is track particles as they go into cells and understand where they go. Now, obviously, a lot of these particles just go to lysosomes. But what we've identified is that there's a cadre of particles that pop off into other interesting locations. Now, you would never see these in normal microscopy because there's too few compared. The reason you see them is that you're only looking for RAB colocalization of events. So you're looking, if you like, at other pathways. To just probe this a little more, be very blunt about this. What I'm showing you here is we've fluorescently labeled the human plasma as green. Please don't worry about that. It works, and we've checked all sorts of things about it. So when you see green, you're not seeing the particle. You're actually seeing the proteins that have been imported into the cell. This is just a lysosomal event. And essentially what's happening here is that a number of proteins are brought into the cell from the external environment. Now, in the lysosome, they're subsequently degraded off, but there's all sorts of interesting things, all sorts of interesting subspecies of proteins that are brought into the cell and across barriers, or at least into barriers. I should be cautious about what I say about across barriers. And so what we're now trying to understand is to see if we can relate how these particles drift into other channels by pulling out the particles and identifying which proteins have been carried in to see if they can be related to the off-site trafficking system. I think that's all I have time for, certainly. Thank you for your attention, and I'll leave that statement with you. And maybe we just need to think a little bit more on where the other particles go and think more of safety by design. Thank you.
Yeah, thanks a lot, Ken. That was a very instructive introduction into this topic. So we have allowance for one question only. Please.
Thanks for your very interesting presentation. And I would like to extend my question beyond of the phenomena that you have described, in the sense that speaking about particles that are circulating, that are designed to target usually a parenchyma. So it is supposed that they have to cross the endothelia, and they will find fenestrated endothelia, so I imagine they can go beyond this endothelia with their own corona. Otherwise, they have to exploit the physiological mechanisms of transport. Then, what do you imagine that will reach the parenchyma? Taking into account that the extracellular spaces, liquids, are devoid of proteins.
It depends on the situation. It depends on the target. It depends a little bit on what the target is bathed in. It depends, for example, on the leakiness of the vasculature and things like this. And it depends on the residence times of the proteins that are carried with you. I think you can't answer that question a priori. You've got to actually study it. What I can say is that it seems to me surprising that if you pull these particles out, just to make it clear, it would not be prudent, based on typical methods, to identify what the proteins are associated. There are just so few proteins, and they're very limited. Yet in situ, very few proteins can have quite dramatic effects. So I don't know if that's a good enough answer to your question, but I guess in general, as particles change compartments, for example, they do exchange some of their proteins. If they get into a low protein environment, they do lose some, they retain others, but those that are retained may still be sufficient to cause modifications to affinity. That, however, was not primarily what I was talking about, by the way. I was not primarily talking about the effect on the targeting, although there is one. To be very clear, what I've shown you doesn't necessarily fix a lot of the problems in targeting, but one thing is absolutely clear. If your particles don't work in the biological environment, if they don't target by the pathway that you advertise in the biological environment of interest, they won't work in vivo. So at a very minimum, I guess what I'm recommending is that we should study these kind of effects in these simple systems.
Okay, thank you again then.